we explained about the anatomy of dicot root, monocot root, dicot stem, monocot stem, dicot leaf, monocot leaf. And next to this, secondary growth. Secondary growth means increase in girth of the plant body due to activities of lateral meristem known as secondary growth. So lateral meristems are the interfascicular cambium and carp cambium. Carp cambium that takes place in the extra development takes place in the extrastellar region, and in the stellar region interfascicular cambium. So once again, secondary growth means increase in girth due to activities of lateral meristem such as carp cambium and interfascicular cambium known as secondary growth. This kind of secondary growth absent in monocots and even in herbaceous dicot plants, except the few herbaceous dicot plants, secondary growth takes place in dicot plants, both in dicot stem as well as dicot root. Let me explain how the secondary growth takes place in the dicot stem. Secondary growth in dicot stem takes place in two regions, stellar region and in extrastellar region. In the stellar region, in the beginning of the secondary growth, in the initiation of the secondary growth starts from medullary rays. This is very important for NEET. Actually, the medullary rays are present between the vascular bundles, parenchymatous cells. These parenchymatous cells, they form the interfascicular cambium, becomes meristematic, and joins with intrafascicular cambium, the cambium which is already present in the vascular bundles. Interfascicular cambium joins with intrafascicular cambium to form a cambial ring. The cells present in the cambial ring becomes meristematic and gives secondary xylem towards its inner side and secondary phloem towards its outer side. The activity of secondary xylem, activity of the cambial ring is more towards the inner side rather than the exterior side. As a result, extensive secondary xylem, this is also important for NEET, extensive secondary xylem can be seen towards the inner side rather than the secondary phloem. And almost the primary phloem and the secondary phloem get crushed because of the extensive secondary xylem, as the activity of cambial ring is more towards the interior side. And during the spring season in the secondary xylem, wider cells are formed. Together constitute, these cells constitute spring wood. Spring wood also called early wood, as it is developed during the early part of growing season. And the spring wood is very lighter when compared to the autumn wood, the late wood. So it is very lighter and very active, low density. So far not asked. So spring wood is very lighter, very active and low density when compared. It has low density when compared with the autumn wood. So during autumn season, narrower cells are formed that constitute autumn wood. Autumn wood also called late wood, as it is developed during the later part of the growing season. Unlike the spring wood, there it is lighter and this is heavier, and active spring wood, this is inactive, lower density, higher density can be seen in case of autumn wood. Spring wood and autumn wood together constitute annual rings. By counting the number of annual rings, we can determine the age of the plant body. That is also very, very important. And in the stellar region, the few parenchymatous cells which lie between the xylem and the phloem and the vascular bundles. They constitute the medullary rays. This medullary rays supply water to the phloem and food to the xylem. This kind of lateral conduction of both food and water is the important function of the medullary rays. So apart from the springwood, autumnwood, medullary rays. So medullary rays are the few parenchymatous cells which run parallelly through the secondary xylem and the phloem. Constitute medullary rays. They help in the lateral conduction of food and water. Apart from springwood, autumnwood, medullary rays. So in the secondary xylem, dark colored hard dead tissue present towards the center in the secondary xylem constitute heartwood. Heartwood is also called duramen. Duramen lacks the conducting capacity and it secretes tannin, resin, gums, and duramen is used in furniture. Hard, dead, black colored tissue present towards the center in the secondary xylem called duramen. Or heartwood as it is present in the center of the secondary xylem. Soft, living, yellow colored tissue present towards the periphery in the secondary xylem called sapwood. Sapwood is also called alburnum. Sapwood or alburnum, heartwood or duramen. These words are also very important for the NEET. Sapwood is alburnum, duramen is the heartwood. These are the changes which takes place in the stellar region of the dicot stem. That is, the parenchymatous cells which lie between the vascular bundles forms the intervascular cambium, and intervascular cambium joins with the intravascular cambium which is present already within the vascular bundles, forming a ring of cambium. This becomes meristematic, gives secondary xylem towards its inner side, secondary phloem towards its outer side. As it is more active towards the inner side, extensive secondary xylem can be seen. As a result, the phloem, even secondary phloem, get crushed. Primary and secondary phloem get crushed. And during the spring season, wider cells are formed called springwood, also called early wood, which has lighter and active and low density. And during autumn season, narrower cells are formed that constitute autumnwood or late wood, which is having high density and darker and then less active when compared to the springwood. Few parenchymatous cells which run parallelly through the secondary xylem and secondary phloem called medullary rays. They help in the lateral conduction of food and water. And the hard dead black colored tissue which is present towards the center in the secondary xylem called heartwood or duramen. Secretion, that is, it secretes, this duramen part secretes tannin, resin, gums and lack conducting capacity. And in the soft living yellow colored tissue present towards the periphery in the secondary xylem, that is called sapwood or alburnum. These are the changes, and sapwood has conducting capacity. These are the changes that takes place in the stellar region. Coming to the extrastellar region. In the extrastellar region, few cells located in the extrastellar region in secondary the stem, dicot stem, may be parenchymatous cells or collenchymatous cells undergo de-differentiation. That means having lost the capacity of divisions, once again they regain the capacity of divisions called de-differentiation. Few parenchymatous cells or collenchymatous cells become meristematic. That region forms cork cambium or phellogen. This cork cambium or phellogen gives phellum towards its outer side and phelloderm towards its inner side. Phellum or carp, phellogen or carcemium, phelloderm or secondary cortex, together called periderm. This is also very important. So phellum or carp, phellogen or carcemium, phelloderm or secondary cortex, together called periderm. Phellum cells are highly suberized. Suberin is present. Even the Casparian stripes in roots, I told you, that is also made up of suberin. Here also, phellum cells or carp cells are highly suberized. As a result, exchange of gases becomes very difficult. In order to overcome this difficulty, the phellogen or carcemium here and there produces thin-walled cells called complementary cells. That is very important. Complementary cells. Sometimes they straight away the people will write, the students will write about the lenticels. No, here the thin-walled cells called complementary cells are produced. These complementary cells break the epidermis, forming an opening called lenticels. Lenticels, lenticels. And these lenticels are also called nematodes. They help in both transpiration and aeration, that is the exchange of gases. And the last point in the secondary growth in the extrastellar region is the important one, that is called the tissue which lies above the fascicular cambium, that is the secondary phloem, secondary cortex, cambium, phellum, all these together called bark. There is soft bark and hard bark. This bark consists of dead tissues towards its outer side and living tissues towards its inner side. And this is about the secondary growth in the dicot stem. Coming to the secondary growth in dicot root. In dicot root, as I told you earlier, pericycle plays an important role in the dicot root. As I told you, the tissues which lie inner to the endodermis collectively called stele in dicot root. Outermost layer of the stele is pericycle. Pericycle plays an important role in the secondary growth of the dicot root. Here also it takes place in two regions, stellar region and in extrastellar region. The cells present in the pericycle becomes meristematic and it forms a dumbbell-shaped ring pushing the phloem towards outer side and the xylem towards its inner side. Thus, increase in girth takes place in the region of the stellar region of the dicot root. In the extrastellar region, periderm development takes place. That means phellogen or carcemium gives rise to phellum or carp towards its outer side, and phelloderm or secondary cortex towards its side, inner side. Thus, the periderm formation takes place. And this is about the secondary growth in dicot stem as well as in dicot root.